Is Jose Manuel Garcia Aznar from Universidad de Zaragoza, who's going to speak on modeling 3D cell motility.
Okay, thank you very much. First of all, I would like to thank the organizer for the invitation to be here in this multidisciplinary conference. I am from the mechanical engineering department, so I had a little bit afar from biology and from mathematics, but okay, I am used to use these terms. So it's clear in this workshop that cell migration is very relevant phenomena for many processes. In this case, it's very important, I think, to take into account the niche of the cellular microenvironment in which the cells are going to be involved. So in my presentation, I'm going to mainly focus on these three level or scale levels. So as mechanical engineering, we're starting to do some simulations in order to simulate some phenomena, but of course these models are very simple and they are perhaps quite simple for modeling some of them. But I think it's very interesting to follow the line of my presentation. So the first of all is how do cells move? They are okay here in this workshop. There were many presentations about that, but I would like to do a simple summary that we can see the cells normally polarized, and later there is protrusion, adhesion formation, and finally contraction and retraction, and the cells move. But as we saw in the morning in the presentation of Professor Witt, we see that the movement is not the same in 2D and in 3D. Clearly there are big differences that later we are going to show. So, which are going to be the main stimuli that in some way are regulating this cell migration? There are many of them, that's chemistry, mechanics, or topography, that can in some way lead this migration. So, as we started to work modeling cell migration, the first approach that we started to use is the most classical approach, using reaction diffusion equations, in which we try to simulate all the scale but from a macroscopic point of view. So we try to simulate extracellular matrix using normally constitutive law, the classical constitutive conservative equations, and later we try to incorporate also soluble factors to how they move inside the tissue, and later we try to incorporate cells. The main difference is cells can exert forces. So we try to incorporate the forces that cells are doing, and the main contribution is to change the equilibrium equation, and we incorporate two terms: the contribution of the extracellular matrix and later the contribution of the forces that the cells are doing. So for our first approach, we try to simulate the forces that are doing the cell in a very simple way. So we try to simulate the cytoskeleton like a set of springs. So in some way we try to simulate this part, the actin and the actomyosin machine, like a contractile system and a spring that simulates the stiffness of the actin. But this actomyosin contractile system is not constant. It's going to follow a similar law that is known in muscles like Hill's law. So there is an optimal slide in which the system exerts maximum force. And later, in parallel, we have a passive spring that is going to, in some way, to simulate microtubules, intermediate filament, the other elements that exert stiffness on the cell. And this approach is very simple, but so we modify the equilibrium forces, and with this simple idea, we were able to simulate durotaxis phenomena in 2D and texotaxis in 2D. Later, we try to incorporate other phenomena in cell, like migration, proliferation, and we incorporate the conservation equation, and normally we use this classical reaction diffusion equation, and where we have here the different reaction terms, and we apply this, for example, to simulate the process of wound healing. How, when we have a wound, the fibroblasts, depending on the growth factors that are released, are going to move to remodel the matrix, and how finally all the wound is completely repaired. So it was a first approach, and our idea is to try to understand how cells are moving. So we try to go down into the scale to understand how one cell is moving, And we use a very simple idea to use voxel finite element approach. So it's very phenomenological law. Our idea was to simulate, in collaboration with people from MIT, we tried to simulate the microfluidics experiments developed in the laboratory of Professor Roger Kamp, in which they apply a fluid flow here, and we simulate one cell like a set of voxels when we have in the middle the nucleus of the cell. And we applied a fluid flow, a difference of pressure, and in this way we can quantify the fluid flow, the concentration of a factor, different mechanical variables, and we propose a very simple model with this a set of voxels where in the middle we are going to have a number of voxels that contribute to the nucleus, and later we incorporate voxels surrounding the nucleus, and all these voxels are going to appear or disappear in function of the local microenvironment. So in this way we consider the voxels that are in the boundary, and for example if we study this voxel, we consider if we have a higher probability to add a voxel here than here, because for example in this case the maximum stress is in this element, so there is a higher probability to add a new voxel there. It's a very simple idea, but with this simple idea okay, we consider the chemistry, fluid condition, mechanical condition, we update the probability function and we add or remove elements. And of course we can also take into account the extracellular matrix degradation. And with this simple idea we can see how the cell qualitatively is able to simulate. This is an experiment in which we have two more cells moving in 3D, and we can see how the cell qualitatively behaves very similar how it's doing in the experiments in 3D. But of course it's a very simple approach.
Sorry,
And yeah,
in the previous slide, it's removing real data.
sorry. Yeah.
This will be
Yes,
from a real system.
this one is real. Yes, the microfluidic experiment. The other two are computational. So later it was a theoretical example. We tried to simulate a scaffold in which the cell is able to move inside with the same idea of the model, and so the model is very simple. We can describe quite well the phenomenon. But of course this approach was quite simple, and we tried to improve this approach to understand how the cell and the fluid are interacting in this way. So we, I don't know why, okay, to understand the role of fluid flow. So the interaction between fluid and cell is very important, but of course the interface in this approach of voxel is very simple. So for this we try to use an immersed finite element approach, where the idea is we have a solid, this is the volume of the cell, so we are going to simulate the constitutive equation for the solid. Later we have the fluid, this is the external, we have the constitutive equation of fluid, and we do a direct coupling. We have in the interface, we have the velocity equality with the velocity of the solid are equal to the velocity of the fluid, and the normal stresses are similar. Later we distinguish, we incorporate the geometry using an immersed surface level set method to incorporate where is the interface, distinguish in this case the fluid and in this case the solid, and we have in the middle the immersed finite element approach, distinguish between the solid and the fluid. So with this simple idea, what we do is we immerse the geometry, we solve the fluid-solid equilibrium at the same time, obtain the new geometry. With the new geometry we update the variables, and in this way we recalculate, and in this way we can simulate how the fluid can deform the cell, for example in a confined environment. We have one cell, in this case we simulate the cell as a solid, this is a fluid, and this is a confined environment, and we can quantify how the cell is going to deform inside this confined environment. So in this way later we can modify the parameter in order to understand how different properties, the level of confinement can influence the deformation of the cell. But of course, the cell normally has a nucleus. So the idea is, using the same idea but with two interfaces, we are able to try to understand how the fluid condition and the confined condition can deform the whole cell, analyzing the nucleus and the external cell body. And later in our interest, we try to go down into the scale to understand how is the cell body. So for that, we started to understand how the cell is contracting. This is an experiment in which one cell is located between two plates. This plate is too stiff, and this plate is less stiff, is more compliant. So they measure that the force that the cell is doing due to contraction change with the time, but depending on the stiffness, they achieve a saturation level. So there is a stiffness in which the cell is not able to distinguish. So the force that the cell is doing is exactly the same. However, there is a level, in this level, that we can see how the cell is able to modify more linearly the forces that the cell are doing. So our idea was to create a discrete particle-based model in order to simulate how the cytoskeleton internally can predict this behavior or can present this behavior. So the idea is to simulate the actin filaments that can be cross-linked with actin cross-linkers, and later we incorporate motors that can walk around these filaments. So for that, we use a Brownian dynamics particle-based model. We simulate actin network with cross-linkers and motor, and we incorporate polymerization from a pool of actin monomers. So later I'm going to describe briefly the model. We incorporate, we use the Langevin equation when we neglect inertial deceleration terms, and we incorporate extension and bending potential and repulsion potential when two segments are very close. In addition, we incorporate the possibility of these cross-linkers can unbind using a bell theory when the force between them is very high. And in addition, this equation is very long, it's better to understand this. When the motor is working here, we use a high speed when the motors tend to walk toward the barbed ends of the filament, of the actin filaments. So with this idea, we incorporate this, a volume of a piece of cell, and we incorporate the external boundary conditions are elastic modulus, and we modify the properties of this elastic modulus, and we are going to see which is the macroscopic response of this network. So we can see that at low stiffness the contraction is very big. However, when the surrounding medium has a big of high stiffness, we can see how the contraction it doesn't exist, but we can see that there is more holes inside the network. If we analyze the mesh, we can see here when the stiffness is low that all the network is together, and we can see how the actin filaments can be compressed and are bending. However, when the stiffness is higher, we can see big holes, and we can see how the actin filament is mainly striped. And the idea is to see what is the macroscopic response of this network. So if we obtain the law, we can see, of course, a temporal dependency, but we can see that when we increase the stiffness, we can see how the forces that the cell or this network is doing is increasing. But you see that when we achieve a level, all the response are in the same place. So we have achieved the saturation level that we observed in the experiment. So this model, although very simple, is able to predict the behavior of the cytoskeleton. So, but in any case, all these models are very qualitative. So our intention is to develop a more quantitative analysis. So we try to do experiments and in addition model in order to try to validate these models. So for that, we try to quantify 3D migration in two different mechanical environments, collagen gels and fibrin gels, and when we incorporate different chemical environments, platelet-derived growth factor and blebistatin. So for that, we are going to characterize these hydrogels, and we are going to combine microfluidics to apply a control gradient, a control chemical gradient, and also numerical simulation to fit the experiments with the simulation. Of course, we have to take into account that, as we have seen here, that two main kind of migration can occur in amoeboid or mesenchymal, and mainly most of the results that I'm going to show are corresponding to mesenchymal cell migration. So if we initially characterize the hydrogel, the differences between collagen and fibrin, and we can see that collagen is more bent than fibrin that is more straight, and we see more or less the pore size is a bit higher in the collagen than in the fibrin. They have a similar radius, and in this case, in the collagen, they are more bundled than in fibrin. We also measure the permeability of both hydrogels. The permeability is the collagen is much more or less 2 times more permeable than fibrin. We quantify using rheology experiments the mechanical properties of both systems, and it's very interesting to observe the following characteristics. The first, both present strength hardening, but collagen presents strength hardening and a strain of 0.5, 50%. However, fibrin presents strength hardening very early, at 10%. And in addition, we evaluate the stiffness, and collagen is 10 times lower, present a stiffness lower than fibrin, about 10 times. So the idea is to use microfluidics to apply a controlled chemical gradient and later to quantify how the cells are moving under this control gradient. So for that, we try to quantify this gradient. So we use microfluidics using the work of laboratory using this protocol, and in this we have 3 chambers. In this chamber, in the pink chamber, we incorporate the hydrogel in 3 dimensions, and later we have a growth factor here, and there is a media factor here. So clearly what we have observed, there are different phenomena. We observe that there are degradation of this chemical factor, but another important factor that we observe is there is diffusion, but also there is part of this chemical factor that is binding to the gel. So we consider that this bound factor can create an apoptotic cue for the cells to move inside the gel. So to quantify the experiment and the model, we use computational simulation, and we are going to use ELISA experiment to measure the amount of growth factor that we have in this channel and the amount of growth factor that we have in the opposite channel. For that, we use a typical equation reaction diffusion that is going to be used in these 2 channels. For this one channel, we use the Einstein equation that is the growth factor is moving free in the channel, and later in the second chamber we have a gel. That's why we have to modify the diffusion coefficient. So for that, we take into account the molecular radius, the radius of the fiber of the matrix, and also the volume of matrix of the hydrogel. So we consider that in the chamber 1 is where occur the maximum degradation, And in the chamber 2, we have the phenomenon mainly regulated by binding. So this is the result that we have obtained, and we can see that there is a good result, and the numerical prediction is quite good in comparison with the experiment. So we assume that in this chamber the hydrogel is going to do two missions. It's going to diffuse, to move, but also the growth factor is going to be binding to the matrix. So this is the gradient that we have observed in collagen that is moving, and this is the gradient of the factor that is binding to the matrix. However, in fibrin we obtain, this is the gradient for the chemical factor that is moving, but there is no binding in the case of fibrin. So after that, we have quantified the migration of cells. So first of all, we analyze using immunofluorescence how the distribution of the protruding, the dendritic structure in both cases. In the case of the collagen, we have multiple branches with active patches. However, in the fibrin is more a fried spindle-like structure. And we are going to study the following case: control. Later we put the cells inside. Later we have cell, but we incorporate platelet-derived growth factor in one channel, and later we add blebistatin in order to know which is the role of the myosin. So we can observe here, this is the movement of cells, and we are going to focus mainly in this area, that is where cells are moving much quicker. This is an experiment of 24 hours, and we can see above all that when we incorporate platelet-derived growth factor, there is a high movement. We are going to study more quantitatively. If we observe the cells individually, we can see that in the control case, it's clearly that in the collagen the cells move much more than in the fibrin. In the fibrin, the cell is creating a lot of branches, but the cell body is not going to move. When we incorporate platelet-derived growth factor, clearly the cell create long branches, but in the collagen the cell move quicker. However, in the fibrin the cell still go on more or less static. It's creating more branches, but it doesn't move. It's also clear that we incorporate platelet-derived growth factor, the migration is directional. When we incorporate blebistatin, we observe that more or less in this case we quantify now, but it's more, the movement is more or less similar, but in this case you can see that the branches are much longer. So we quantify all of these experiments, distinguish three zones due to the different gradient. We distinguish the zone that is close to the factor, a second area and a third area, and we quantify the mean speed and the effective speed. So if we see here, it's a first picture of all results. We can see clearly that in the control, I'm going to detail now all the results, but clearly when we incorporate the growth factor, clearly increase the migration, and it's clearly increasing the collagen, but not in fibrin. In fibrin, all the time, more or less the movement of cell is static, is not going to move. If we quantify in detail, we can see here that when there is no factor, the migration is not directional, and we can observe that it's higher in the case of collagen in comparison with fibrin. In the case of fibrin, when we incorporate the growth factor, we can see that protrusions increase, but okay, the migration increase, but it's very, very slow increase. Okay, it's very low increase. However, in the case of collagen is where we can observe the highest increase. There is clearly a directional motility, but if you see the cells that are in red are the cells that are in the area close to the growth factor. So clearly they are moving more, we can observe here, than the cells that are in the second and third area, where the chemical factor is far away from the chemical factor. So the difference is, the difference is quite big, as we can observe. In fact, in the third zone, the speed is very similar to what we obtain in the case of the control. In the case of blebistatin, when we compare blebistatin in the case of fibrin, the results are more or less similar. And in the case that we incorporate blebistatin with the growth factor, we can observe that the migration doesn't increase, it's reduced. Okay, although we can see that above all in the first area, in the area when we obtain more binding factor and more chemoattractant factor. So this is the part of the experiment that we have done, and later we have tried to create a simple model to try to reproduce these experiments. For that we have used a discrete approach. So the idea is very simple. Our model will have this kind of behavior of the cell, so we are going to represent the cell center, and later we have to create different kind of branchings, and we are going to incorporate three main mechanisms: chemosensing, later philopodia growth, contraction and retraction and protrusion, and later we incorporate the constraints of the extracellular matrix. So first, the chemosensing, we assume the cells are embedded in the extracellular matrix, and we assume that all the receptors are distributed equally over all the cell surface. And we assume a chemical reaction using a Poisson process, using a very known algorithm that is called Gillespie algorithm. In function of this algorithm, we are going to estimate the areas when we have a higher probability than a protrusion, a branch is going to appear. So normally we incorporate a reaction diffusion equation that we obtain at higher probability. P3K is going, 3K is going to, sorry, sometimes I'm speaking English in Spanish, is going to produce, and this point is going to create the philopodia protrusion. And this protrusion, to understand the mechanical effect on the matrix, we are going to consider like an inclusion of Archer-Randall matrix using the classical theory of Edelweiss. So using the Edelweiss theory, we can simulate the growth of the philopodia like an increase of temperature, or the retraction of the philopodia using a decrease of temperature. And using the formula of Edelweiss, we can simulate the stresses on the extracellular matrix. So in this way, the analysis is very, very quick. So we have this scheme, we have the chemical factor, the cell, and how the extracellular matrix is deforming. In this way, we have the cell nucleus, we have a number of branches, and in the cell nucleus, we consider the addition of all the forces that the branches are doing, and we consider the viscosity of the extracellular matrix. In this way, we can evaluate the speed of the cell speed. So we have done some preliminary simulation, and we can see how the cells are moving in function of how the branches are growing and they are contracting. As I show you, there are preliminary results, and we can see here the trajectories in both cases, in collagen and fibrin. Of course, it's much easier to analyze the influence of different concentrations on how they move. And of course, in the experiment, we only have currently one experiment, but we can see that in vitro, our model is able to predict quantitatively the result of the speed migration of the cells. And also, we have quantified the size of the philopodium in the case of collagen and the case of fibrin. So, okay, I'm going to finish on time. So my main conclusions are, okay, I have tried to present several samples to show how numerical simulation can help us to understand some phenomena in cell migration. We have to keep in mind that numerical simulation presents many limitations. Reduce impact if we don't have validation, and in my opinion require the combination with other types of analysis. Here we have some of them, combination of experiment and image processing for trying to find a systematic validation of the numerical results. I think it's very important also to quantify, to move from mechanobiology to mechano-chemo-biology, because sometimes when we only study mechanics or we only study chemical, we are losing part of the phenomena. So I think always that we try to look at all the phenomena, but also combining mechanics and chemistry. I think it's very important. And also, although numerical modeling is a promising tool, there are many limitations. We still have many limitations. And that's why I would like to finish saying that, in my opinion, the development of novel numerical strategies that allow to link scales and reduce the computational cost would be very useful. And also, would be useful tools that could be easy to be used for other people, not only for people that are used to using simulation. Then, okay, this work has been developed thanks to the European Research Council and the Spanish government and local government, and of course it is part of the work of two PhD students, Ollana and Federico, and also part of my group has worked in this presentation, and of course it has been done thanks to the collaboration with Professor Roger Kahn from MIT, that he taught us to work with the microfluidics, and he has collaborated in the development of some of this model. With Tae-Yoon Kim from Purdue University, we have been collaborating in the model, a discrete model for cell contraction, the cytoskeleton. Also, Thomas Ruber from Austria has been working in the merged finite element approach. Johann Meyer has helped us with the measurement of rheology experiments. Miguel Vicente Manzanares helped us with the interpretation of resolvable force with blebistatin, and also Carlos Ortiz in the microscopy analysis. Thank you very much.
So questions? Yeah. I was wondering if you could connect your earlier model. You showed many models, but earlier you showed some sort of cells moving in 3D that actually indeed did not show those protrusions. And is there any connection with that model that you showed maybe one of your first slides to this model, where you acknowledge the presence of those protrusions?
Yes, okay. In the first model that we developed, we have really not many idea how works the migration in 3D. Okay, so later when we started to do experiment, we have realized that at least in the experiment that we have observed, the migration is more like protrusion and dendritic structures that is different what we initially have understood reading in bibliography. So the first model, it was more phenomenological, and the first analysis that we observed is more focused on tumor cells, and it seemed that it was similar, but when we quantify really how was the migration, we quantify doing measurements more quantitative. Okay, the first model we are going to go on using, but not for this kind of cell movement. So what I can say is, when the first experiment that we observed, we didn't see this kind of structure. It's true that in this kind of gel, it was a fibrous gel. I think the kind of extracellular matrix can also regulate the shape of the cell. So perhaps the shape of the cell is is depending on how is the extracellular matrix, and that's why I think both models can be valid. Okay, but one model is more focusing matrices that are fibrous, and the other model I think is more valid in a structure where we don't have a matrix that is fibrous. Okay, I think it's my main idea, but.
Yeah.
So, I'm interested in the difference between fibrin and collagen here with your cells. They don't move in fibrin, but they move in collagen. So is it just because they don't make the right protease to degrade fibrin, or is there something else going on?
Yes, this is a question that we have. We don't have clear. It's a question that the cells are not able to degrade fibrin, or if they are not able to deform. We don't know it. In fact, our next idea is to try to understand what is happening. But in other works, we have read that the cells normally are able to degrade fibrin. But in this case, there are skin human fibroblasts. But what we were surprised because it seems that they are not able to degrade. So we would like to investigate more about this because it was surprising for us. And of course, I don't know, but probably we're going to try to block metalloprotease, but I think if we block, it's going to move less. So I don't know, so we are thinking how to proceed in this direction. Because it really surprised us.
Other questions? May I have one, actually. You have made a case that you need to bring the models and the experiments very close together. So how do you actually calibrate the parameters of the models against the experiments? Do you just trial and error, or do you have a systematic procedure like applying the tools of I don't know, primary calibration? And
I would like to say that we have a systematic way, but it's not true. Okay, what we are doing is normally the speed of the models is completely different than the speed of the experiment. Normally, my main background was computational, so now I am incorporating more people with experimental experience. My interest is the students can do the same student. I would like that they can do the experiment and the model. But it's very difficult to find people that like to do both things. Normally they prefer everything experiment or everything model. So when I think that I have the impression when a person is doing both things is better because you achieve a systematic validation. When a person is doing the experiment and another person is doing the model, it's more difficult. I try to do my role and intermediate, but normally the experience is we are waiting for the results. After that, we try to see what happened in the experiment. We measure, we do prediction, and later we repeat again the experiment.
It's kind of a trial and error.
Yes, this is the idea.
I have a question about your voxel-based finite element model, because I was wondering, so how does it compare to similar voxel-based models, like the cellular plots model,
Yeah.
And what are advantages relative to other models?
Yes. Okay. I never work with plot model because my background is finite element analysis. So I can say that at the end it's very similar, but the results at the end I think is very similar. It's a different tool, in my opinion. But I don't have really experience on plot model. My experience is in particle-based model, or more that is more mechanical. For me, plot model is different. I don't have experience. Okay, I have read, but I never work as a user with plot model, so I cannot really say nothing specific about that. Sorry.
Okay. Any other question? Okay. So if not, let's thank the.
Okay, thank you.
Exactly on time.